My name's Stephanie Wills. I'm a DPhil student at Oxford, co-supervised by Frank and Charlotte in the Oxford Protein Informatics group. Today I'm going to be talking about algorithms for progressing crystallographic fragment hits at XChem, and specifically I'll be talking about this tool that we've been developing, which performs fragment merging using the Fragment Network. So just to provide a quick overview, I'll give a really brief background into fragment-based drug discovery and fragment-to-lead progression. I'll talk through our published work, which describes that tool that I mentioned, a bit about our current work, which looks at developing this tool further and expanding the chemical space that we're using to find mergers, and then how we're starting to deploy some of the design algorithms that we have at XChem for active campaigns within the ASAP discovery project. So just to start, fragment-based drug discovery, as opposed to high-throughput screening, involves the screening of very small molecules, so molecules that typically have a molecular weight of less than 300 Daltons. They have very low molecular complexity, so typically bind with weaker affinities in the high micromolar to low nanomolar range. So several advantages of using such small molecules is that we typically get higher hit rates because they bind more promiscuously. They cover a greater proportion of chemical space, but in particular, because we have a smaller starting point, we have greater control over further synthesis and optimization of properties such as affinity, which we can do in a step-by-step iterative process. So the question that we want to answer is that once we have these fragment hits, what can we actually do with them? So what we want to do is look for methods that, using these data from the fragment screens, is able to identify compounds that recapitulate some of the atoms and substructures that we see in the original fragments. So we do this using structure-guided optimization, and the purpose of this is to increase potency, maximize the number of interactions, and to begin to elucidate an understanding of the structure-activity relationship for a target. So there are three main approaches for fragment elaboration. The first is fragment growing, where we grow fragments to reach new parts of the pocket. Fragment linking, where we link together two distinct binding fragments using molecular linker. And fragment merging, where we have two adjoining or partially overlapping fragments, and we want to find compounds that incorporate substructures from each of them. So at XChem, which is based at Diamond Light Source, we perform fragment screening using X-ray crystallography. And this is really powerful because if we have crystal structures or fragments bound to the protein, this allows us to confirm the exact binding pose. We can map possible interactions within the pocket, and it can allow the structure-guided optimization of the fragments into larger, more potent molecules. So the elaboration techniques that we've been focusing on are more of the fragment merging and linking techniques, because as you can imagine, these can be really efficient and powerful approaches for increasing potency. And merging it's been shown in some XChem projects to lead directly to on-scale affinities. So these are some examples for a few different targets. For example, SARS-CoV-2 Mpro. We found that mergers that were designed for fragment hits that were overlapping in the pocket led to compounds that had IC50s in the low double-digit micromolar range. So what are the types of existing approaches that we have for identifying follow-up compounds following these fragment screens? So a lot of examples of fragment mergers in the literature have been manually designed by expert medicinal chemists. So this is obviously a really useful and powerful tool for developing these compounds. However, it's quite difficult to scale up. You can't necessarily exploit all the possible opportunities, all the possible enumerations of fragment pairs in the pocket. So we want to look at developing more automated approaches for doing this. So there are some de novo design methods, things like deep learning and generative models, which are also really powerful, but a lot of these techniques, even the state of the art, can struggle with synthetic accessibility. So some of the compounds, they might be really difficult to synthesize or very expensive to do so, or they might not be possible to synthesize at all with the current tools that we have available. So a way to circumvent this is just to look directly at commercial catalogs of compounds, because we can very rapidly and cheaply identify follow-up compounds in order to rapidly progress these fragment hits into something more interesting. So sort of classical ways of doing this are using things like similarity or substructure searches, but there aren't a whole lot of formalized workflows for how to do this, and also these aren't necessarily the most computationally efficient techniques. So they'll typically involve calculating similarity against an entire database of compounds, whereas we want to do this much more efficiently. So the way we can think about this in terms of this diagram is, in the center, we have compounds that we can get really easily, things that are in the catalog that we can, that are very synthetically accessible. And we have the compounds that actually make sense for the target. So what we want to do is to improve the efficiency with which we sample this area of accessible chemical space. So this leads me on to our paper, which looks at performing fragment merging using the Fragment Network. And we published this in JSIM earlier this year. So first of all, the Fragment Network is a concept that was first described in 2017 by Aztecs Pharmaceuticals. And this is basically a graph database representation of chemical space. So in graph databases, we have nodes that are connected by edges, and edges represent relationships between the different nodes. So in this database, we represent molecules as nodes, and the edges represent transformations between the molecules. So the way we generate this is, if we have a molecule, we can iteratively remove substituents, rings, and linkers in order to generate all these further connected nodes. And then when we populate the database with several compounds or compounds from several commercial catalogs, this provides a really nice way of probing the chemical space around a compound of interest. So we can look at neighboring nodes where substructures are either being added or removed, and it provides a much more sort of chemically intuitive way of traversing chemical space. So the version of the Fragment Network that we re-implemented at XChem contains compounds predominantly from enamine, but also some from Moldport and ChemSpace. And the version that we used when we published this paper had just over 120 million compounds in it. So the aims of this work are, can we use the Fragment Network in order to find purchasable fragment mergers that maintain substructures of the crystallographic fragment hits, and thus hopefully we'll be able to recapitulate the binding pose and interactions of the parent fragments. And we also wanted to compare this technique to the more traditional similarity search in order to find whether the Fragment Network has any actual additional use beyond the sort of more standard techniques. So to do this, we performed the search using the Fragment Network and one based on a similarity search to find fragment merges using our database. And we looked at 4 different test systems. So these represent 4 XChem screens, so fragment screens for which we have the hit data across a few different targets and organisms. So we have SARS-CoV-2 main protease, we have this helicase protein, NSP13. We also looked at P. gingivalis peptidase and human PARP14. So in addition to running the pipeline for these targets, we also performed retrospective analysis using some example data in the literature for mergers that were manually designed, and have experimental data so we could see whether our pipeline was actually able to pick up things that we knew bind. But I'll talk a bit about that in a bit more detail in later slides. So this is quite a complicated slide, and I'll take you through step by step, but essentially this shows the entire pipeline that we used for querying the Fragment Network. So essentially we start with the crystallographic fragment hits. We query the network in order to find merges in 2D, and then we use some filters in order to get that list down to something more manageable, and hopefully the most promising compounds that we think will adopt the pose of the parent fragments. So yeah, for each of these targets, we start with the crystallographic screening data, and we enumerated all pairs of compounds to merge. And we do some basic filtering here, removing pairs of compounds that are quite far apart or might be too similar. We then queried the database in order to find these fragment merges. So the way we do this with the Fragment Network is we start from one of the fragments. This forms our sort of seed fragment in the query. We find the node that represents this fragment, and then we make up to a couple of hops away from this node in order to generate some diversity. With the other fragment, what we do is we decompose it into its constituent substructures. And then we can make hops away from these compounds that then incorporate one of the substructures from the other fragment, thus resulting in compounds that contain substructures from both of the original fragments. And this is an asymmetric query, so we can reverse this for the fragment pair. As I mentioned, we compared this against a more traditional similarity search. So similarity search is conducted using molecular fingerprints. So the molecular fingerprints are essentially just a really simple vector representation of a compound, where each of the numbers represents the presence or absence of a certain substructure. And it's quite easy to calculate, thus, similarity between two molecules using these vector representations. So what we do is we represent all the compounds in the database with these vectors, and then we calculate similarity of the fragments to all of the compounds in the database, and we pull out things that have a similarity above a certain threshold to both of the original fragments, thus hopefully representing what would be fragment merges. So after that, we have all these 2D proposed merges, and what we want to do is to filter them and to find the things that are most promising and are most likely to recapitulate the binding pose of the original fragments. So we pass them through some sort of simple and cheap 2D filters, things like ruling out things based on Lipinski rules or ruling out things that look like maybe expansions of just one fragment. Then we pass them through the more computationally intensive 3D filters. So the main one that we use is conformer generation. So what we want to do is to generate a conformer for the merge that reflects that of the original fragments, because we want to see, is it able to bind in the same way and thus make the same interactions? So to do this, we use Fragmenstein, which is a tool that was developed by Matteo Ferla. And essentially what Fragmenstein does is just to place the compound using the atomic coordinate space from the fragments. And it does this by looking at substructure matching between the fragments and the merge and looking at the positional overlap, and then performs minimization within the protein structure. So what we can do then is we can rule out compounds that aren't able to adopt the same conformation. Those that aren't able to do it in a physically reasonable way, we can filter out those that also don't fit the binding pocket because of clashes with the protein. So we ran this for the 4 different targets, and what we find is that each search technique identifies comparable numbers of filtered compounds for each target. But what we also find was that these two techniques seem to be quite complementary. There doesn't seem to be a lot of overlap between the compounds that are being identified by the 2 different search techniques. So these plots are T-SNE plots, and basically these are just low-dimensional representations of chemical space. They just to show the chemical space that's occupied by the filtered compounds in 2D. And filtered compounds identified with Fragment Network are shown in blue, and those identified by similarity are shown in orange. And what you can see is they seem to be operating in quite distinct and different regions of chemical space, which seems to show that they seem to be quite complementary. These plots show for each target, for each fragment pair, the number of filtered compounds that are being identified by each search technique. The Fragment Network again in blue, and similarity search in orange. And one of the things that you can see, which is quite striking, is that for a lot of fragment pairs for each target, one search technique seems to identify filtered compounds while the other doesn't, and vice versa, which again seems to support this notion that they're complementary and seem to perform better for different types of merging opportunity. We then looked a bit more detail at what the different types of merging opportunity the 2 different search techniques are suited for. So to do this, we classified the merging opportunities according to the degree of overlap between the 2 parent fragments. So here we have in orange and purple merging opportunities where the 2 fragments are very overlapping in the volume. And then the second one is where we have partially overlapping fragments, where that partially overlapping structure is a ring structure. So the hypothesis for merging is generally quite obvious or quite clear. The 2 other categories seem to represent sort of more difficult merges in that there's not an obvious hypothesis for merging. So the third one is partially overlapping merges where there is no overlapping ring structure. And the fourth is where we have fragments that are completely non-overlapping. So these represent more linker-like opportunities, except we're not incorporating the entirety of the 2 individual fragments. So we consider these to be more difficult because there's no sort of obvious hypothesis for which substructures within the fragments to incorporate and the connectivity of how to do so. But what we find is that Fragment Network is a lot more efficient at identifying emerging types 3 and 4, while the similarity search identifies more of type 1. So again, suggesting that they seem to be better performing at different types of tasks. So we also performed a retrospective analysis using literature examples of manually designed merges as test cases. So this first test case looks at compounds that were proposed through the COVID Moonshot project. So this project crowdsourced designs for the fragment screen that was performed against SARS-CoV-2 main protease. And we use this because within this data set, there are examples of manually designed mergers. This is one example, this submission, this tri-uni submission of compounds, where we have these 5 overlapping fragments, and the compounds that were manually designed as mergers of these fragments, several of them have IC50 values, so provide a nice test case for us to see whether the fragment network is able to retrieve them. So what we did was run the fragment merging pipeline on these 5 fragments and then compare the results against compounds with experimental data in the COVID Moonshot project. So what we find is that the fragment network is able to identify this known binder without performing the full R group expansion. This binder has an IC50 of 59.6 micromoles. However, it doesn't have a crystal structure, so this is the Fragment-Stein predicted structure that came out of the pipeline. We do also find that the fragment network identifies several very close analogs of known binders against Mpro. And as you can see, the Fragment-Stein predicted structure, which is shown in white, closely mirrors that of the crystal structures, which are shown in cyan. And a lot of these compounds have low double-digit micromolar IC50 values. We do also perform some additional R group expansion on the compounds, which is then able to pick up even more known binders, but I can talk about that later on. So we also performed another test case, which looked at manually designed mergers in the literature against M. tuberculosis protein ETHR. So the authors had these 2 fragment hits shown in blue and green that each bind in 2 different conformations, and they manually designed these mergers against them. And they're particularly interested in these 2 because the crystal structures were shown to overlap well with the parent fragments. So what we did was to run the fragment merging pipeline for these 2 fragments and then generate posters based on all the different combinations of fragment conformation. So we find that the fragment network does identify the known binder compound 4 after filtering, and as you can see, the Fragment-Stein predicted structure in white again mirrors that of the crystal structure shown in cyan. The authors of that paper also designed several analogs of compound 5, of which we're able to identify several very close analogs or matched molecular pairs. So this example shows the fragment network identified structure in white and the crystal structure in cyan, and they only differ by the single nitrogen atom. But we wouldn't expect the fragment network to be able to pick up this compound anyway, because the original nitrogen atom wasn't found in the original fragments. However, this compound had an IC50 value of 22 micromolars. So to summarize, we provide a flexible pipeline for finding purchasable follow-up compounds for crystallographic fragment hits using merging and linking-like strategies. We find that the fragment network is able to identify mergers that maintain exact substructures of the parent fragments. And it seems to be that the fragment network is complementary to a more traditional fingerprint-based similarity search, so that we argue that these 2 techniques could be used in parallel, and we could use the fragment network to increase the productivity of the catalog search. The retrospective analysis shows that the fragment network is able to identify potential binders against 2 known targets. And also this represents a potentially more computationally efficient approach for searching catalogs, as opposed to doing, for example, a virtual screen or similarity search where we have to do a calculation against every single compound in the database. The fragment network instead restricts the search to the compounds in the neighboring chemical space around the fragments of interest. So definitely have a speed up in the computational resources required. So next, I'll give a bit of an introduction to some of the current work that we're doing in expanding this tool and developing the sort of second iteration of this fragment network search. So some of our motivations are, how can we maximize the chemical space that's explored by these fragment merging techniques while still remaining faithful to the parent fragments? We find that the first iteration of the fragment network merging pipeline fails to find catalog-based mergers for certain pairs of fragments. So what we want to do is to try and improve the productivity and hit rate by instead looking for mergers that might recapitulate interactions or pharmacophores seen in the parent fragments without incorporating exact substructures. And we also want to see if we can do this in a computationally efficient way. So what we're searching for is what we've termed imperfect merges. So if we consider the first iteration of the pipeline looks for perfect merges where we incorporate exact substructures, we're instead looking for things that might replicate pharmacophores while still maintaining the binding pose and interactions that are being made by the parent fragments, but might not contain exact chemical structures. So this shows sort of the early version of the pipeline that we're using to try and test this. So we start off with our fragment hits, and instead of enumerating pairs of fragments to merge, what we're doing is enumerating compatible substructures from that fragment pool. And we make sure that compatible in terms of things like their spatial arrangement, and we can also do things like prioritize substructures that are responsible for some of the most observed interactions or sort of the most important interactions. We can then query the network in order to find these imperfect merges. So what we do is this time, instead of specifying that we want the merges to contain exact chemical structures, we can replace some of the substructures with things that are pharmacophorically similar using on-the-fly similarity calculations. We then end up with these proposed 2D merges or 2D imperfect merges that we can then generate conformers for. So while our first conformer generation method relied on matching substructures between the fragments in the merge, we instead using the pharmacophore coordinates in order to generate conformations for the merges, and then perform additional minimization after using Pyrozetto or OpenMM. So we've tried running this and are developing this on some of the projects that we're doing. But this is just an example of some of the kinds of compounds that we can get out. Given that we have 2 fragment hits and substructures that we want to merge, we can end up with what we call perfect merges that incorporate exact structures from the original fragments. And then we can then begin to replace substructures with things that are pharmacophorically similar or can replicate some of the interactions, or replace both of the substructures in the final merges, thus increasing the diversity in the final pool of compounds that we have, but hopefully still maintaining the conformations seen in the original fragments. So I'll just give a very brief description of how we're trying to sort of deploy some of these fragment elaboration algorithms at XChem right now. So XChem is part of the ASAP Discovery Program, which is an NIH-funded center focusing on developing antivirals for 3 viral families, which consists of many projects, which I won't go into huge detail now, but XChem is focused on Project 2, which involves deploying automated structural biology and fragment screening against viral targets, then conducting analysis and designing compounds and fragment expansions of some of this fragment screening data that can then be further screened and sort of inform this iterative cycle of design. This would then allow development of these target enabling packages, which can then feed into later stages of the pipeline. So within this, we're sort of trying to focus on this aim 4, which is this compound design step, and trying to establish a process for this. And this is really useful for people like me because it enables prospective validation for design algorithms and also provides a platform for method development. So the first target that we are looking at doing this for is the enterovirus D68 3C protease. So the fragment screen for this was led by Ryan Lithgow, and he actually has, he presented a talk at this forum, so please check that out if you want to learn more about the target. But essentially we screened over 1,000 compounds against this target. And for design purposes, we focused mainly on fragment hits against the catalytic site, which exists on monomer A. So against this site, there are 18 fragment hits that predominantly bind in these S1 and S2 regions in the pocket. So this provides quite nice merging opportunities because we can try to find compounds that bridge across these 2 pockets. So this just shows sort of an overview of the process that we're establishing for getting compound designs ordered, which we can then perform further screening on. So we're doing this at the moment for enterovirus D68 3C protease. And what we do is at XChem is we've started with all our different design algorithms. So things like looking at shape and color, This is where my work fits in, so looking for enumerations that are based within the catalog, and then also some other members are deploying some methods where using more generative models, but then trying to find analogs within the catalog that we can then rapidly purchase in order to allow rapid progression of these fragment hits. The compounds from these methods are then subjected to multi-factor ranking using things like shape and color scores, which look at the pharmacophoric and shape overlap with the parent fragments, looking at the types of interactions that are being made, looking at the RMSD, and using these to whittle down the list to a short list of compounds that can then be passed to medicinal chemists, who are then able to sanity check and curate this into a purchase list. So this is, we sort of established a process for this with a few design iterations for this protease, and the compounds are then ordered from Enamine, and then further screening is performed on them. And then these compound designs are then put onto Fragalysis, which is the online platform that we have where this fragment screening data is available. So our very first iteration, which was just sort of a very quick rushed iteration just to establish the logistics, I think it took maybe about 6 or 7 weeks to get from compound design to new structures. So there are 3 hits from that, which are shown here and are found on Fragalysis. But we have a lot more data incoming from further iterations and targets. So hopefully this will provide some nice validation for some of the tools that I described today. And then these compounds can then further feed into more work at XChem that's being developed, which looks at performing SAR exploration from these follow-up compounds. So in summary, the Fragment Network merging tool provides a pipeline for finding mergers from the catalog. And we show initial development on an updated pipeline that's looking at expanding the chemical space that we explore to find these mergers using the pharmacophores. And these ASAP projects have helped us to start to develop a workflow for ordering these follow-up compounds and on a rapid timescale to allow rapid progression of these fragment hits and help provide some validation for the design algorithms used at XChem. So thanks to everyone else who worked on this, everyone at ASAP and my supervisors, and thank you for listening.